Hello, this is Gina Piscatelli with the third lecture for A&P2 Week 1. In this lecture, we'll talk about the composition of plasma. And this picture shows, on the first slide anyway, shows normal clear plasma as well as plasma that's obtained after someone has eaten a fatty meal. Pleasant, huh? Okay, so let's recall first the components of blood. 55% by volume is plasma, 45% will be formed elements. When we draw someone's blood, collect it in a test tube, and then spin it down in a centrifuge, like this diagram shows, the cells generally sink to the bottom. And the cells that are heaviest and sink to the bottom are the red blood cells. So we call this region of red the hematocrit, right? So the hematocrit is typically around 45% of blood volume. We'll talk about that again. And then the plasma is at the top because it doesn't weigh as much. It's not as dense, I should say. Now in the middle, though, is this kind of fluffy region. That's called the buffy coat. And that's where the leukocytes and the thrombocytes usually end up. Okay, so if you're collecting plasma, you have to be careful not to disturb that buffy coat with your pipette if you want a clean sample. Okay, let's go to plasma composition in a little bit more detail. Just plasma now. Plasma is 90% water. Within that solvent, water, aqueous environment, there are proteins. By volume, 8% proteins. Most of these proteins, if not all, are made by the liver. Not all, because we digest and absorb amino acids, of course, but 8% of the proteins are made by liver. One is albumin. It is the most abundant of those proteins, those liver proteins. It is a carrier molecule for things like hormones that need a carrier molecule. It also is really important in contributing to the osmolarity of the blood. So you need to have solutes in order to keep the water in the blood so it doesn't, by osmosis, leave the blood vessel. So albumin helps greatly with osmolarity of the blood, as do any solutes, but albumin contributes the most. It's a rather big protein. There are also globulins present in plasma. These globulins are antibodies. Now, globulins aren't completely made by the liver. The antibodies are made by B lymphocytes, but there are other globulins that are carrier molecules that are made by the liver. And the third type of protein that we'll become familiar with in this class is fibrinogen. So fibrinogen is an inactive protein, but when activated, it becomes fibrin and forms a clot. So those are the proteins that we find in plasma. Also in plasma are other dissolved solutes like electrolytes, ions, sodium, chloride, potassium, calcium, bicarbonate. Some nitrogenous wastes, mainly from breaking down the proteins that we eat. Urea has nitrogen in it, uric acid, creatinine, which comes from our muscle tissues, and ammonia are all nitrogenous wastes. Typically, the kidney will filter out these nitrogenous wastes from the blood, but there's always a certain amount in the blood. Because we talked about the transport of gases by blood, you know that there are gases, but they're found in the plasma as well as in cells, but we're just focusing on plasma now. So there's dissolved oxygen in the plasma, dissolved carbon dioxide, and dissolved nitrogen. Also in the plasma are nutrients like glucose, amino acids, fatty acids, vitamins, cholesterol, you name it, nutrients, and hormones, which we talked about in A and P1. Okay, let's talk a little bit more about the importance of these plasma proteins like albumin, fibrinogen, globulins, and any other solute, to tell you the truth, like amino acids and glucose and things like that. It's really important to keep a certain osmolarity of blood to prevent the loss of fluid or water from blood to the interstitial fluids. When we do lose fluid from our bloodstream out to the tissues, we notice swelling. And the term for the swelling of tissues is edema. And you can see a picture of this person's foot on the bottom. The ankle looks swollen and the foot looks swollen, but we also can call it pitting edema, P-I-T-T-I-N-G, because a pit forms if you press your thumb against that swollen tissue, like right there. And if the pit stays for a while, the depression stays for a while, we call it pitting edema. Now the picture on the right just shows you interstitial fluid exists between cells, and that's where the fluid from the blood would go into the interstitial space. Now, how do we prevent edema? How do we prevent fluid loss? Well, it's by having plasma proteins. So the osmolarity of the blood will attract the water, and so it will stay, the water will stay in the blood. If instead someone has hypoproteinemia, that means low protein in the blood, low plasma protein concentration, they get diseases associated with that, not just swelling, but they can get liver disease, kidney disease, malnutrition, starvation, and severe burns. Meaning that's what causes hypoproteinemia. Sorry, I didn't make that clear. One particular disease associated with hypoproteinemia that I want to mention is kwashiorkor. This occurs in individuals that have an adequate caloric intake, so they eat enough, but no protein in the diet. So protein malnutrition. In that case, without enough protein circulating in the blood, right, the liver isn't able to make up the difference because it doesn't have the amino acids to work with. So it can't make the proteins. And we see swelling, and I'll show you a picture of this. So here's some children with kwashiorkor on the left, and they have a diet deficient in protein. So they don't have protein for their muscles to grow. Their muscles atrophy. And if you look closely, you'll notice the arms and legs are quite skinny. There's not a lot of muscular tone. But what's interesting is that, yes, there should be swelling, right? But what's interesting with prolonged low protein intake is that the swelling accumulates in the abdomen. We call this kind of swelling ascites. So these children are exhibiting ascites and muscular atrophy, their limbs and things, due to a diet deficient in protein, like they're just eating rice or something. That's a little bit different than just plain old undernourishment. So I'm going to go back to the previous slide. Now, obviously, someone who doesn't have an adequate diet in general, quantity-wise, is also deficient in protein. But so you see undernourishment in all the different categories of nutrients. So you'll see then low energy and weight loss. This condition is known as marasmus, and I'll show you a picture of that. So it's a little different. You don't see ascites in the abdomen. You see a wasting and overall emaciation and loss of subcutaneous fat. Both conditions are serious, of course. Now, sometimes, since we're talking about plasma, sometimes students ask me, what's the difference between plasma and serum, right? Because, I mean, there's plasma donation and there's serum donation, that kind of thing. Well, the difference is that plasma has clotting factors in it, and serum has those clotting factors removed. So plasma has clotting factors in the plasma, including fibrinogen and calcium, all of which are needed to form a blood clot. So that means when you collect blood, you spin it down, and you have this plasma here on top, you could still get a clot unless you add an anticoagulant. Okay, so its shelf life is shorter. Okay, you have to use it right away, the plasma. Serum, on the other hand, won't clot when left standing because the serum is collected after a clot has formed. So they can collect the blood like normal. They usually don't spin it down right away. They just let it sit at room temperature. Then they spin it, and the clot is usually down at the bottom. So this will have the calcium, the fibrinogen, and all the clotting factors, and the serum is up here. Okay, so when do you use each one? Like, when do we need each one? It depends on how fast you're going to use it. Oh, but first let me show you what a blood clot looks like. Okay, so here's a damaged vessel on the bottom. And if a blood clot did not form, we would lose blood cells as well as plasma to the interstitial space, right? But here we have a blood clot forming, and you can see those yellow threads around all the red blood cells and the white blood cells. So that's a blood clot. The thread is fibrinogen, and I wanted to show you a real picture. This is an microscopic, scanning electron microscopy image. Ugh, I can't talk. And it's a very small vessel. And all the red blood cells are stuck to the fibrinogen threads. There's also white blood cells and platelets in there too. So that's what a blood clot looks like, up close and personal. And blood clotting happens by a couple of different pathways, which we'll talk about later. But we have these clotting factors in our plasma. They're just named with numbers. Clotting factor 12, 11, 9, 10. Here's 6. Here's 7. We got a lot of clotting factors, right? This thing called prothrombin, which is a precursor to a clotting factor. Fibrinogen. We have all these things. Ultimately, to get a clot, a cascade of reactions has to happen to produce those threads of fibrin. That's the take-home message. There's a lot of different reactions that have to take place. And for the reactions to take place, you're going to need calcium. So I circled calcium at a couple of steps here. And so if you remove calcium, those steps can't take place, and you won't get fibrin forming from fibrinogen. You can't get a clot. So often to treat plasma, they'll put a calcium clotting agent in it so that it won't form a clot when it's left on the bench top. Okay, so what are the advantages and disadvantages to using plasma versus serum and stuff? So as we know, if you want to keep it around for a while, you're going to have to add an anticoagulant of some kind. But if you're going to use it right away, it's nice because you can obtain it really quickly, right? You don't have to add an anticoagulant or wait for it to clot and just collect the serum. A bad thing, though, is when you try to pipette this out, you could contaminate your sample with the white blood cells and platelets that are at the buffy coat region, but usually people get pretty good at that and they just don't go down that low. So that's not usually a big deal. So plasma is less stable for storage. Serum is more stable for storage because the clot has already happened. No clotting factors are even present in the serum because it was collected after a clot formed. So you wait 30 minutes for clotting, then you collect the serum and you get a clean sample. That's the difference between serum and plasma, or plasma and serum. That's not in your study questions, but students always ask me, so I thought I'd put that in. Thank you very much. That's the end of this lecture.